Okay, so hi everyone. My name is Alex and I'm a PhD student in Matthew Harks's lab, which is split between the School of Chemistry and the UK Dementia Research Institute. And today I'll be talking about studying protein aggregation in Parkinson's using single molecule microscopy. And throughout this talk, I hope to give you some indication as to how and why we study this and its relevance for understanding Parkinson's. So firstly, I'll give some background as to why and how we study aggregation before going in depth into my two studies on this subject. So Kiani has already mentioned some of this, but just to reiterate, the pathological hallmark of Parkinson's is the presence of abnormal aggregations of protein called Lewy bodies in the midbrain. The major component of these bodies is aggregated alpha-synuclein, which is a small disordered protein, and as Kiani mentioned, like a wiggly line, which is highly prevalent in the brain. However, its main function is unclear. This is the basic structure or sequence of alpha-synuclein. The details aren't important for this talk, but for your reference, changes occur in this area of the protein in inherited familial Parkinson's. Notably, aggregated alpha-synuclein is found in both sporadic and inherited Parkinson's, and as such, alpha-synuclein aggregates are key targets of research for studying the fundamental basis of Parkinson's. So what do I mean by aggregation? It's the process when a protein changes from its normal functional shape as a single monomer, and then begins to clump to form these small oligomeric aggregate structures, so these oligomers. And then it keeps clumping together to form these larger fibril structures. When studying aggregates, typical bulk experimental methods which measure the change in an aggregated sample as a whole have previously been used to get an overview of this process. However, they don't give information about individual molecules within a sample, meaning that we can't fully understand the finer details of aggregation. Also, these aggregates are not very prevalent in a sample. So if I make a comparison between our aggregated sample and, say, a crowd at a football stadium, it's like trying to look for individual people in a noisy crowd with no specific way of picking them out. So we need to use what are called single molecule techniques to enable us to identify individual aggregate species. And these are a set of microscopy techniques that use fluorescent molecules or dyes to detect and analyze these individual molecules. The key message here is that in this project, we use these techniques to improve our understanding of how alpha-synuclein aggregates might be involved in causing Parkinson's. So I thought it would be useful just to show you the microscopes that we use so you get an idea of what we work with. On the left, we have what's called a TURF microscope, and on the right, a single molecule confocal. Essentially, both use lasers to illuminate fluorescent molecules or dyes attached to our protein of interest. And I've included an arrow of where the sample would be illuminated for your reference. So for the TURF microscope, this is essentially just a high-tech microscope that allows us to pick out individual molecules in our sample and gives us an image. So like this example image of alpha-synuclein aggregates, which are the little white structure things here. And then from analyzing these images, we get information about the individual molecules within them. Secondly, the single molecule confocal also picks out individual molecules, but instead gives us a photon count. So in this example, larger bursts of photons, so larger peaks here correspond to larger molecules that are in your sample. And then we analyze the different peaks, the different counts that are coming through to get information about them. So my first study involves looking at a specific inherited variant of alpha-synuclein, and this formed part of a larger study with University College London. So studying variants is important, as although most Parkinson's cases are sporadic, the variants are good models for studying different processes in Parkinson's. So A53T is an early age onset, rapidly progressive variant of the protein, which comes from a change in the genetic sequence right here. And we wanted to solve both how and where it aggregates in the cell to understand its pathology better. So for studying this, we first added labeled protein to ourselves. So if you think back to the stadium crowd analogy, think of this as making people in the crowd and marking them out by making them wear a colored hat. So if you add a hat to one half of the people you want to find and a different colored hat to the other half of the people, when they group together or aggregate, we see the two colored hats together. We then collect the aggregates produced in the cells at different time points, and then we look at these with our single molecule microscopes with the aim of determining how A53T aggregates in cells. We first visualize these aggregates on the TURF microscope. So we first overlapped images taken of the same field of view, but with different fluorophores or hats illuminated, so that we confirm that they're overlapping in the same molecules and that the aggregates are composed of the two different hats or fluorescent molecules. We then estimate the size of these aggregates by tracking the decrease in their fluorescence over time. So essentially over time, as a fluorophore is exposed to the microscope laser, it undertakes chemical damage and can no longer fluoresce. If we liken it to the hat analogy again, it's as if the hats individually lose their color over time, and then we can no longer detect them. So as you kind of see in this little clip here, from tracking how many little molecules or hats fail over time, we can determine aggregate size as the total number of molecules or hats that disappear. And then we get a graph showing the size of these aggregates over time. So here for these A53T aggregates, most are quite small, and the size range doesn't really change much over time. Moving on to confocal data, which gives us information on the range of the aggregates that are produced. We did this for a different alpha-synuclein variant as well, the A30P variant, just for a comparison. And we show our data using what are called aggregate heat maps, which are essentially 3D plots where each hat or molecule is a dot on the map and is placed depending on where they fit. So from the dots, we can see both the range of the aggregate size on the x-axis and how closely packed they are on the y-axis, which is important as more compact oligomers or aggregates are typically more toxic. Additionally, the color coding is similar to contours on a map of a mountain range, with increasing color from blue to red indicating higher numbers of aggregates. So this data shows that A53T in both cases here shows more aggregates and a wider range of aggregates than the A30P here, as you just see far fewer dots placed on the map compared to here and here. And again, we see that most of the aggregates that are present are smaller in size. So this is similar to the data we saw on the previous slide. Therefore, the key finding here is that A53T aggregates rapidly in cells. Following from this, we want to see where in the cell the A53T alpha-synuclein aggregates. So again, labeled alpha-synuclein is added to the cells, except in this case, using our colored hats, we are essentially trying to find what section of the football stadium they're located in. So similar to before, we produced heat maps for where the aggregates are found in cells, and found that the mitochondria are a particular hotspot for aggregation. So mitochondria are what produce energy for the cell, so the aggregation here damages the energy-making process, leading to toxicity and further aggregation of alpha-synuclein. Overall then, this study provides us with a detailed model of how alpha-synuclein aggregation begins in the cell in Parkinson's. I'll also briefly cover a second study which I'm currently working on, and is looking at how alpha-synuclein exists and aggregates in the brain. So in real life, in the body, alpha-synuclein gets this chemical cap here called an acetyl group in a process called acetylation, as Kiani mentioned earlier. However, until now, most studies have looked at the protein without this cap, so there's a lack of data, especially single molecule data, on how alpha-synuclein would aggregate in the body, which is almost like trying to see how good someone is at tennis without giving them a proper racket. So we want to study this effect in normal alpha-synuclein as well on the G51D variant, which occurs up here, similar to the A53T variant. And it's similar in that it causes early onset and aggressive Parkinson's as well. Therefore, our aim is to see how alpha-synuclein aggregates naturally in the body with its acetyl cap using our single molecule techniques. And in this study, instead of labeling our protein directly, so like with a hat, we use a dye called thioflavin T instead, so that we see the protein in its most natural state. And this dye specifically binds and illuminates aggregates, which we detect on our microscopes. We looked at the differences in aggregation for both standard, by which I mean non-variant protein, and G51D, both with the naturally occurring cap or a tennis racket, and without, and by then splitting these aggregates into different sizes. So if you look at this figure for reference for the small sized aggregates that we get, it shows a comparison of the standard alpha-synuclein in blue and the G51D variant in red, and then with the cap in the dark colors and without the cap in the lighter colors. With the bars, we can then see the numbers of aggregates on the y-axis, and then their increasing levels over time on the x-axis. So we don't see huge differences between the different proteins with the smaller aggregates. However, when we look at the medium and especially the large size aggregates, there are significant differences. And the addition of the cap seems to have an almost inverse effect on aggregation for the standard G51D variant, especially when looking at these large aggregates here. So we see that standard uncapped protein and capped G51D produce far more larger aggregates than capped standard protein and uncapped G51D. This is very important as it was previously thought that G51D aggregated more slowly than standard protein, but when we study how the protein exists in the body, the opposite appears to be true. Therefore, this has implications for our current understanding of the aggregation process in the body. We also see differences between the variants when you visualize them on the turf. So these representative images are taken after 120 hours, so near the end point of the aggregation process, and there are noticeable differences in the numbers and structures of aggregates produced. Also, for your reference, the scale bar here is 5 micrometers. In comparison, a human hair is between 20 and 200 micrometers thick, so the width of this entire image is about the width of a human hair.
Alex, you've had 10 minutes.
Okay, thank you. In the normal unacetylated protein, so without the cap, and in the acetylated G51D, we see a lot of fibrillar structures compared to the other two variants here. So in summary from these slides, acetylation or the cap has a significant effect on aggregation of these alpha-synuclein variants. So finally, just a bit of outlook and overview. So far this study gives us a basic view of how the acetyl cap and the G51D variant affect aggregation. However, we now want a more physiologically relevant context. So currently our collaborators in University College London are doing some cell toxicity tests by adding aggregates to cells to determine what effects the variants have. While here I've started testing the interaction between alpha-synuclein aggregation and cellular membranes. So I mentioned before that alpha-synuclein's main function is unclear. However, it's known to be involved in regulating synaptic vesicles, which essentially are little molecules which are used to transmit chemical messages or neurotransmitters from one neuron to the other in the brain. We can make a synthetic model of these vesicles in the lab, which are called liposomes, and they look a little bit like this. And we can then use these to model interactions between alpha-synuclein and the vesicles that they regulate. In summary, with this work, we'll get a better understanding of how the G51D variant and the acetyl cap in alpha-synuclein affect pathology in Parkinson's. This concludes my talk. Thank you all for listening, and I'd like to thank the people who are involved in this research, as well as the funders who have contributed, especially Dr. Jim Love, AR UK and UCB, who have been giving us continued support throughout this project. Thank you very much.
Thank you, Alex. Could you stop sharing your screen now?
Yeah.